by this thing. Okay. We call it the Houston Obesity Nutrition Research Center. And then this is a special lecture that we organize this year. And then it's very generously sponsored by our sponsor, Sino Biological. Right? And we're very grateful for their support to make this event happening so that we can have Professor Sadaf Farooqi to visit us in Houston and then give this lecture. So before we invite Sadaf to the podium, and then let me invite Dr. Burgess to come to the podium to promote their product.
Thank you, Dr. Shu, and good morning, everybody. As mentioned, I'm Rob Burgess, and I head up business development for Sino Biological. I am here with a few of my colleagues today. They are out in the lobby here with a nice table set up. These include Lauren Monshine, Shebna Massey, Priscilla Hernandez, Rajleen Gogar, and Siddharth Ardza. And we are honored and privileged to be the sponsor of today's seminar by Dr. Farooqi on obesity and other metabolic disorders. So I'm just going to give you a little brief overview of Sino Biological, try to keep it under 5 minutes or so, and then turn it over to Dr. Farooqi. So our company was founded back in 2007, specifically as a spin out of MIT, and it was spun out of the late Dr. Daniel I.C. Wong's lab, and Dr. Wong and his student, Dr. Liangxi Shi, were experts at the development and mass production of recombinant proteins, primarily in mammalian cells. So HEK293 cells, CHO cells, etc. The reason why you would generate a recombinant protein in a mammalian cell is very obvious. They tend to have the proper post-translational modifications in three-dimensional structures and really just look as natural and in vivo-like as possible, in contrast to proteins made in bacteria, in yeast, and maybe even the baculovirus insect cell system. So the company was spun out. Headquarters was located in Beijing, China, and we have since grown those headquarters to 110,000 square feet of laboratory and office space. And a couple of years ago, we actually went public on Chi-Next, which is a Shenzhen Stock Exchange subsidiary. And we've grown our company now over the last 15 years or so to almost 1,500 employees. About a third of these have upper level, either master's or doctoral degrees, primarily in the life sciences. And our primary offering at Sino is catalog-based, primarily recombinant proteins, as well as recombinant monoclonal and polyclonal antibodies. And we actually generate about a third of our services through the implementation of custom contract research services as well for the generation of proteins and antibodies. And I'll just say a little bit more about our company. So we have direct sales in North America as well as different parts of Asia and Australia. We sell into about 100 countries worldwide, both direct and through almost 40 distributors. And we topped about $240 million in revenue in 2020 at the height of the COVID pandemic. Many people don't know this, but about half of all the antigen-based COVID diagnostic kits that were on the market had cytobiological antibodies in them. So we have 10 physical sites worldwide. We just opened new labs in Houston, Texas, right here in Houston, and that's specifically the C4B, which is right down the road on the corner of Holcomb and 288. And this is where we specialize in custom CRO services. And that is for the manufacture of very difficult to develop and produce recombinant proteins and recombinant antibodies. So just a little bit more about our offering then. Sino is a one-stop reagent and services provider for recombinant proteins and recombinant monoclonal and polyclonal antibodies. We actually have the world's largest selection of full-length bioactive recombinant proteins made in mammalian cells at almost 9,000 unique targets. And we have chosen some of the best-selling proteins as well as strategically chosen drug targets. And we've manufactured about 15,000 antibodies which bind to those. We also have about 700 or so either ELISA kits or validated antibody pairs. And we sell the genes as well. For every protein and every recombinant antibody, we can simply subclone those into your vector of choice and have those shipped out to you. So really, this represents, I think, very well Sino's offering. We could take a cDNA sequence from you, and we can generate it into a full-length recombinant protein verify the bioactivity of that, and then generate recombinant monoclonal or polyclonal antibodies or antibody pairs to that target if you would like. This is our citations, example of our citations. We have over 10,000 citations as assayed by PubMed, and it's growing at about 2,000 new citations for our products. So it's still growing roughly exponentially. And our laboratories in Beijing are ISO certified 9001 for documentation control, 13485 for medical device certification. Ironically, proteins and antibodies are considered medical devices, and our labs are specifically certified by the World Health Organization in Beijing as being GMP compliant. Our labs in Houston here are all brand new, but we are pursuing all of these certifications for those labs as well. And I just want to mention a few obesity and I think type 2 diabetes related targets that we have currently available at Sino. That's the beta isotype of the thyroid hormone receptor for liver disease, on the GLP-1 receptor, which is actually, it's my understanding, it's the receptor for the world's most famous anti-obesity drug, Ozempic, which is also known, I believe, as GLP. And then the GCG receptor, which is another drug target for type 2 diabetes and obesity. And so with that, I just want to thank everybody for their attendance and thank the organizers for the opportunity for Sino to host this wonderful seminar today.
Thank you. Thank you also for the support. Right, so now I will introduce our guest, Dr. Sadaf Farooqi. Today, really, I don't think we really need a long introduction. If you Google her name, there's tons of videos, and then you can find her Wikipedia page, and then you will learn all about her. Just a really few words. Dr. Farooqi is from University of Cambridge. She is a professor in medicine, and also sees patients. So she received her medical training as well as PhD training in UK and then stayed there. And then she is, I guess, very well known in the field of obesity, especially the genetic basis for human obesity. There is perhaps 20 and 30 very well known monogenetic reasons for human to develop severe obesity. You can name it. And then Dr. Sadaf Farooqi's work is behind those gene names. And then you will appreciate how much contribution she's made to this field. And then as a very corporate recognition for her work so far, and then she has numerous awards, and I just named a few. And I believe 3 years ago, she was elected as a fellow of the Royal Society. And then that's, I think it's equivalent to the member of National Academy of Science here in US. That's a huge deal. And also, I was fortunate to actually at her award reception about 4 years ago at San Francisco, where the American Diabetes Association gave her this outstanding scientific achievement award. That's also a very big deal in the field of diabetes and obesity. And then actually part of the reason that we were able to have Sadaf Farooqi to visit Houston this year is because she is going to receive the Clinical Research Award at the Endo Society, which is in Boston in a few days. And then so I asked her whether she can spend a few days earlier with us, and then she kindly agreed. And also we have generous support from Sino-Biological to make this happen. So I can keep going, but I will give the podium to Dr. Sadaf Farooqi, and welcome.
Thank you very much, Young, for the very kind and generous introduction. Great to be here today. I must admit that coming to Texas from Cambridge, UK, makes me feel very, very small and being dwarfed by these huge buildings and fantastic facilities that you have here. So from my small little base here in Cambridge, we've been doing research over a number of years now into trying to understand the mechanisms that regulate human body weight and their relevance to obesity. And you'll see that we've done some work in people, in cells, and in mice. And of course, we have a long-standing collaboration now, 10 years, with Yong Chu and his team. So you'll see some of the work that has resulted from our collaborations. So of course, when we're thinking about the clinical problem of obesity, which is really the starting point for me, we know that obesity, defined as an excess of body fat that adversely affects health, has really changed dramatically over the last 30 to 40 years. And that rise in the prevalence of obesity, led of course by highest prevalence rates being here in the US, is driven strongly by environmental factors. The amount of food we eat, the type of food we eat, and the fact that it's easily and cheaply available. There's also a huge link with socioeconomic deprivation. In many countries around the world, it's the people with the least financially well off who actually have the highest rates of obesity in children and in adults. And essentially, it's our environment that is tipping the balance between the amount of energy that we take in and the amount of energy that we expend, leading to weight gain. But it's not quite as simple as that, because within a given environment, there's a lot of variation in body weight. Some people develop obesity, some people are at high risk of obesity, and others can eat what they like and they stay slim. And we now know that the explanation for that variation is down to genetic factors. How do we know that? Well, actually, some of the earliest evidence came from studies of twins. So if you look at identical twins, you can see from these photographs that actually the identical twins are very similar in body weight, in body shape, and body size, whereas there's a lot of variation in the non-identical or dizygotic twins. If you look at twins who are separated at birth and live at completely different environments, despite that change, they have a remarkable similarity for their body weight as adults. If you look at children who are adopted and they live in a different environment, they actually share weights with their biological parents rather than the adoptive parents with whom they share the childhood environment. And very elegant studies undertaken by Claude Bouchard in Canada really demonstrated how our genes influence our weight when they basically studied identical male adult twins for a whole year in a research study, overfed everybody by 10%, so people gained weight, but the amount of weight people gained was very similar for two members of a twin pair and very different across the sets of twins. So our genes will influence how we respond to the environment. And altogether, if you take any two people in this room, 40 to 70% of the variation in weight is down to variation in our genes. So our genes play a major role in our weight. How do our genes do that? Well, they operate right across the body weight spectrum. As you'll hear, many of the genes that we and others have been found by studying people with severe obesity. But we also know that thinness is highly heritable, something that we've demonstrated, and that genes act right across that spectrum. I think one of the things to clarify is, and sometimes there's a bit of sort of misunderstanding about this, is there are different types of genetic variation. We all have genes that influence our weight. There are many common genetic variants in the population. Some of those have a very small effect on a person's weight, maybe 1 or 2 kilos over a decade, why somebody's much more likely to gain or less weight. And these can be added up together to make something like a polygenic risk score. That's not really what I'm going to focus on today. What I'm going to focus on today is the other major genetic contribution, which is relatively rare variants, but they have a major effect. So if you can understand how that rare variant has an effect on body weight, you get a much clearer idea of the major drivers of this system. So I'm going to talk mostly about rare penetrant genetic variants. Now, how do these genes work and how do they influence the physiology, the system that regulates our weight? Well, we've known for about 100 years that the system that regulates our weight must be based around the hypothalamus, because if you lesion the hypothalamus in an animal, or actually you have a child with a tumor of the hypothalamus, those individuals will gain a lot of weight, develop obesity. So something in that center of the brain is critically regulating weight. We know that the hypothalamus is critical for regulating many other things. So it regulates temperature, blood pressure, reproduction, growth. And so it's a critical part of the brain that receives signals from internal signals from the body and external signals from the environment, and then coordinates a set of responses to maintain homeostasis. And those responses are regulating hormone secretion, autonomic function, or indeed behavior. And essentially, what I think is this useful model is depicted on the lower panel here from John Speakman, where for many decades we've thought about a set point, and that's clear in animals. If you fast an animal, it will lose weight. If you release the fasting, it will eat more to regain the weight and get back to where it started. That's a set point. Human beings don't quite do that, actually. If you take volunteers or medical students, as we generally do, you starve them and you calorically restrict people, they will sort of lose weight. But if you release the starvation, people will eat more to regain the weight, but they don't just go back to their immediate set point, they overshoot. Okay. So in human beings, of course, if we had a perfect set point after Christmas, when people have eaten, or Thanksgiving, have eaten lots and lots of food, and then they stop because they've gained weight, you'd think you'd go back to the set point, but of course you generally don't. And that's because we probably have like a range through which we oscillate. Okay. And likely explanation is that our genetic factors influence where your range lies. So there'll be some people who generally have a higher range, and they will tend to go towards overweight or obesity, particularly in a given environment. But at other times they may swing their weight down. And there'll be other people who just have a lower BMI range. And I think that's a useful way of thinking about how our genes and biological mechanisms influence our physiology. Now, of course, the first sort of molecular underpinnings of how this system works came from the work of Jeff Friedman and colleagues in 1994, who basically studied these obi mice, severely obese mice that you can see, an inbred strain that were found to be missing the hormone leptin. And that hormone leptin was shown to be coming from adipose tissue, acting in the brain to regulate appetite and weight. And if you give these mice that are lacking leptin back leptin injections, you can reverse their weight to normal. And that really proved the primary role that leptin is having, and really led to the way of our understanding of this system, whereby hormones from fat, such as leptin, act on the brain, specifically on the hypothalamus and also on the brainstem, to maintain energy homeostasis, as we were talking about. You also get signals from the gut, short-term signals in relation to meals, hormonal signals and neural signals that tell you about fullness after a meal. Those are often received in the brainstem, but then there are links between the brainstem and the hypothalamus and other brain regions. So that work was really has really been unraveled through studies in mice, but we were able to show that this system is also relevant to human physiology through the study of these children who now, 25 years ago now, we identified as having mutations in the gene that encodes leptin, so the equivalent of those obi mice. And so these were two children, as you can see, with severe obesity. They were of Pakistani origin, born in the UK, otherwise completely well, no developmental problems, and they had undetectable levels of leptin. Just for the youngsters in the audience, this was my first ever assay in the lab, and I was pretty convinced I'd done something wrong because there was no leptin in any of these samples. So then obviously I did it again, and in fact, same happened again with lots of control. So undetectable levels of leptin in the blood, which didn't make any sense, because normally you make leptin from fat. Children have severe obesity. The more fat you have, the higher should be the leptin. But actually they had no leptin. And the reason for that is they had a homozygous mutation in the gene that encodes leptin. So they didn't make any of the functioning protein. So that was the first evidence of a single gene disorder causing human obesity, and also proof that leptin is important for human weight regulation. But of course, we knew that we could give leptin to the mice and correct their obesity. So we then started a clinical trial giving injections of leptin back to these children. And I'll show you on the next slide the effect of that. But essentially, through the study of these children with leptin deficiency pre and post, we've learned about leptin's physiological role in humans. And so what we learned is one of the major things that leptin does, it affects appetite. Okay. So in the absence of leptin, these kids, the first thing that I saw was they're incredibly hungry. They always want to eat food, and they're constantly asking for food. And any kind of food is exciting for them, even English hospital food, which is obviously a disorder of appetite. Okay. And to be honest, that was quite an important finding, because up until that point, everybody assumed that our appetite, what we want to eat and choose to eat, is entirely down to voluntary control. You choose what you eat and what you do, right? All of the obesity literature until that time was talking about metabolism, slow metabolism. Is that why some people develop obesity? So this was really critical to changing our understanding of actually, you know what, appetite has a biological basis. And so we found that these kids eat a lot, but the other thing that they do is that they really liked any kind of food. So I joked because they really did like hospital food. Okay, so this horrible, plain looking food that looks like something you wouldn't even give, to your worst enemy. But they would get excited and start, looking excited and happy that this food was being delivered to them. And I didn't really understand what that meant. I knew it didn't make any sense. Only some years later did I realize that this was about reward. Okay, they were finding food rewarding, whatever the food is. And so we thought, how could we test this in a human being? And just around that time, this technology was being developed using functional MRI. Okay, so it seems like a long time ago now, but essentially you do MRI scan, you get anatomy of the brain. Okay, but if while someone's lying in the scanner, you subject them to a task, you can see which parts of the brain light up or are activated by that task. So if you're a dementia researcher, you would get people doing a memory test when they're lying in the scanner, and then you see which parts of the brain do and don't light up. So what we did, and it hadn't been done at that point, so we had to figure it out ourselves, was do a simple task of showing the kids pictures of food versus not food as a control. Okay, and what happened is when we showed them pictures of burgers and cakes and everything else, scored 10 out of 10. Even when we showed them pictures of broccoli or a raw egg, that scored 10 out of 10. Right? So any picture of food, it's a highly abnormal response, caused a big activation in certain key areas of the brain, the striatum, the nucleus accumbens, and areas like that. And then when we treated them with leptin, that response dramatically went down after just 7 days. Okay. And the response in the brain was linked to response in their behavior. So they also started to turn away food and become much more choosy about the food that they eat. So essentially, we could show that leptin has a major role in regulating appetite and also in regulating food reward. It also affects immune function and also the timing of the development of puberty. So without leptin, these children don't go through puberty, and with leptin, they go through puberty at the appropriate developmental stage. And so this is the sort of just the photograph, which I like to show, which really, I think, tells a million stories. But essentially, this is the dramatic effects of leptin treatment in 2 children that you can see here. Really life-changing for these children by understanding the mechanism driving their obesity and then treating them effectively. And it is predominantly about affecting food intake. So, of course, fast forward over many years, we and others have then looked at the pathways in the brain that are downstream of leptin action. And so leptin goes and acts in the hypothalamus on neurons, and there are many, many neurons, but our sort of focus as a field has been on the initial primary neurons that respond to leptin and other circulating signals. And those include AGRP neurons, which are there primarily to tell you to eat and prevent you from starving. And that's really the fundamental role of this system, to prevent you starving. And then you also have neurons that express POMC, or proopiomelanocortin, whose major role is to tell you to stop eating after a meal. Okay, and the POMC neurons release melanocortin peptides, which are agonists at the MC4 receptor, the melanocortin 4 receptor, on second-order neurons, and AGRP is an antagonist of that same receptor. So it's a very neat system that regulates appetite and eating behavior. And what we've shown over many years is that disruption of this part of the system, the one that's normally there as the brake on eating, will lead to severe obesity. So I won't be going through every gene, but I just want to sort of tell you sort of how parts of the puzzle have come together. Is that downstream of leptin is POMC, this neuropeptide, which is cleaved into the smaller melanocortin peptides, which bind to the MC4 receptor. And children who lose POMC, or the enzyme that cleaves POMC, will also develop severe obesity. It's interesting that we've identified that many of the genes that we found converge on this critical pathway. It's not multiple different pathways. There will be connections to what is probably a major central circuit. So one of the ways that we've done this is that we've recruited a large cohort of children called the GOOSE cohort, Genetics of Obesity Study. It sounded funny when I was a PhD student. I'm still using the same name now, 30 years later. But essentially, what we thought was, where are you going to find the genes that are regulating weight? Genetic disorders often present in childhood. Rather than looking at people who have had a lifetime of environmental exposure, look in childhood, look in the more severe or extreme phenotype. Okay. And so we recruited predominantly children with the help of many colleagues around the world, and then studied their DNA and found mutations in many different genes, which ended up converging on those same POMC neurons that you can see here. And so one example, this is work that we did collaboratively with Yong's lab. In fact, he had already figured out that this molecule SRC1 modulates the function of these neurons by interacting with STAT3, which is downstream of the leptin receptor. And then we said, hang on, we've got human mutations in SRC1, and then we worked collaboratively to show that those mutations affect that interaction and then POMC transcription. And effectively, it modulates, it's almost like the kind of amplifier of the leptin melanocortin signal. And here you can see in some very elegant studies from Yong's lab that a mouse model of the human mutation gains weight on a high-fat diet, and as well as showing reduced POMC transcription, you see reduced firing of the POMC neurons from that mouse model. And this work was really key to establishing that these genes are causes of obesity and potentially treatable, as I'll show you shortly. But you can see a couple of other examples here of genes that seem to all map onto this pathway. The other thing that we learned is that it's not just genes that function on this pathway, but maybe you can change the development of these neural circuits. So my colleague Sebastian Bourret in Lille and also UCSF was working on a family of axon guidance molecules. So the semaphorin 3 ligands, receptors, and co-receptors, which are known to be involved in brain development. And they basically act as guides, telling the neurons where to go in different parts of the brain. And in the hypothalamus, he had showed that they affect the development of these POMC neurons from their starting point to their kind of final point. What we did was we found that actually patients, or in our goose cohort, had mutations in all of these different components, the ligands, the receptors, and the co-receptors, and that in cells, we could show that they affected the guidance function of these neurons. And then we made a mouse model of disruption of the neuropilin 2 receptor, and that had a reduced density of neurons in those particular populations. This is just another example of we're learning there's a lot of complexity in this system, which would make sense. It's a system that's there to prevent you starving. It needs to have fairly robust core, but lots of ways of fine-tuning or modulating that system. This is an example of the first sort of non-coding RNA, which Markus Stoffel in Switzerland had found to cause obesity when it's disrupted in the brain, and then showed that it was down to the disruption of those same melanocortin neurons and its downstream targets. But the key to this is the melanocortin 4 receptor, which comes downstream of those early signals. And this is the gene in which we find the commonest genetic cause of obesity, 5% of our cohort, and actually 1 in 300 people in the population have mutations in the melanocortin 4 receptor that impair its function. Now, it's a G protein coupled receptor, so it's been an important drug target for obesity over many years. And what we did was we functionally characterized many of these mutations in cells, and we also characterized the patients. And what we learned is that, again, the patients eat a lot because MC4R is that brake to tell you to stop eating after a meal. The brake doesn't work, so you eat more. But really interestingly, we saw a genotype-phenotype correlation. So the worse was the mutation in cells, the worse was the patient's clinical phenotype, and the more they ate at a buffet meal. So this kind of work really led a lot of interest in pharmaceutical companies who were then targeting the MC4 receptor to try to see if they could develop some drugs. One of the things that we can do with the discovery of these genetic conditions is that the patients provide us with the opportunity to understand the role of that molecule in human physiology and disease. And we've been able to work side by side with colleagues who work in mice to say, okay, well, you're figuring this out. Does that apply in humans? We observe this in humans. Can you identify the mechanism in mice? And that's been a very nice way to be able to work and advance the field. So here's a quite cool example of that. So this leptin MC4R system in rodents, there's quite a lot of data showing that it's important not only in how much food you eat, but also in macronutrient preference. So do mice want to eat fat, carbohydrate, etc.? And in fact, there was very good data showing that essentially if you disrupt this system in some way, you get an increase in fat intake at the expense of sucrose, whether it's genetic disruption or pharmacological inhibition. So we thought that's very interesting. Might we have the same in human beings? There's no real evidence about fat preference in human beings to date. So we designed a study to try to test that. So that's quite a challenging thing to do because humans don't eat pellets of food, and our food has got lots of different components in it. So what we designed was this dish here on the top right. This is a very traditional English dish called chicken korma and rice. Okay, and what we did is you designed the meal so that you could vary the fat content, but everything else looked and tasted the same. So you have either 20%, 40%, or 60% fat, but people can't tell. You do a big sensory discrimination test where you randomly mix them up and you see, people can tell which is which and they can't tell. Okay. You have to do some fancy things like change the cooking temperature and the oil and this kind of stuff, but the ingredients are all the same. And what we found is that when you present people with a large tray, in the UK, it's considered a large tray. I think it's kind of Texan size, but you have a sort of large tray of low, medium, or high fat chicken korma, and you see what people do. Okay, and compared lean people, people with obesity, and people with the MC4R mutations. And what happened is that everybody ate about the same amount of food, the total meal content, but people with the MC4R mutation ate 95% more of the high fat food. So this is some early signal in the mouth, oropharynx, or when they're eating, because by the time they've eaten the meal and the gut hormones have kicked in, it's done, that made them want to have the high fat food. Okay, so this is very fascinating and aligns with the work in mice. So then we thought, okay, well, is it the fat, or is it the fat that's very rewarding or palatable? So then we need something that's also rewarding. Well, that would be sugar. So then we did a high sucrose test. And here we needed a dessert, which you could handle changing the sucrose. And so this is also a very British food called Eton mess. Okay, so a bit like Cheesecake Factory. So basically you go low, medium, high sucrose. And what happened is most people prefer the sweeter food, okay, which kind of makes sense, but not the people with the MC4R mutation. In fact, they really didn't like it. In fact, sometimes they even refused to do the test. So actually they don't like sucrose. And what was interesting is that there wasn't really much published work in mice at the time we did this study, but actually it was published just as pretty much as we were ready. And again, the same happens in mice. So MC4R deficient mice don't like sucrose. So what does this mean? Well, I think what it means is this pathway is there to defend against starvation. So the people who have genetic disruption of this pathway are always hungry, they like any food, they behave as if they're starving. So what we're seeing is they have increased hunger, they want to eat food, they have increased food reward. Any food is rewarding to you if you're starving, makes sense. And actually, if you're starving, it would make sense that you prefer fat, because you get twice as many calories per gram of fat, and also you can store excess fat if you were going to eat it, rather than carbohydrate, for which you have a limited storage capacity. So I think this is telling us about very fundamental pathways that affect our eating behavior. Okay, and then just really following up on the point about how genes can affect our understanding, they can also provide us with new insights to develop drug targets. So MC4R was our sort of favorite gene, as you can see here, and we did some recent work with well, no, not so recent, a few years ago with Nick Wareham in the UK Biobank. So there's a large cohort of 500,000 people who volunteered for research, and we studied all of the variants in the MC4 receptor, 61 different variants, characterized them in cells. And what we found was loss of function variants in the population. We kind of expected that. And the people in the population who have those were more likely to have obesity, and that fits with the children with obesity. But we also found gain of function variants, variants that make the receptor work very well, and they were associated with protection from obesity and type 2 diabetes. And why did they do that? Well, they do it through two different cellular mechanisms. They basically keep the receptor on the cell surface for longer. So you either impair the internalization or you accelerate the recycling. And so the receptor stays on the cell surface, continues to give the signal to break on eating, so you don't feel so hungry and you're protected from obesity and diabetes. And the way that these mutations work is they preferentially guide signaling through the beta-arrestin pathway. And so there's now a number of companies working on targeting these mechanisms for new therapeutics. But one of the lessons sometimes is that if you observe a human phenotype, it can guide you as to thinking about a mechanism which can then be tested. But sometimes we expect to see things and then we don't see them, and that can also be a clue. So one clue here is that the people with the MC4 mutations that we were seeing for all our different studies often didn't have high blood pressure, which is what you would expect for people with obesity or severe obesity. In fact, their blood pressures were quite normal and often on the low side. Okay? And what we found is that they certainly have a lower prevalence of hypertension and lower systolic blood pressures. And why does that occur? Well, it occurs because they have impaired sympathetic tone. Clinically, these folks are very chilled out. Okay, they look very laid back. I used to think it must be my excellent bedside manner, but then I realized it's impaired sympathetic tone. So we can't really explain that. And then the way we measure that is that you do what's called a clamp, which is you infuse insulin into someone who's not diabetic, and so you can drop their blood glucose, but at the same time you infuse glucose in the other arm, and you work out how much glucose you need, which gives you an index of insulin sensitivity. So that's a strong physiological stress signal. And normally people's heart rate goes up when you do this. But in these MC4R patients, the heart rate goes up much less than in equally obese controls because their sympathetic system is not activated. Now, this was important because at the same time that we were doing this, a number of companies had developed drugs targeting MC4R, and while those drugs caused weight loss, unfortunately, they increased blood pressure. So inhibiting MC4R decreases blood pressure, stimulating MC4R with a drug increases blood pressure. So sadly, that led to the closure of those drug development programs, but it did show a tight link between this pathway and autonomic function and the regulation of blood pressure. So that's something we followed up many years later with Michael Cowley in Australia, with some very elegant studies in mice, showing here that if you high-fat feed mice, diet-induced obese mice develop high blood pressure, but not the mice that are lacking leptin, the leptin receptor. Even when they're fed on a high-fat diet, they don't get high blood pressure. When you give leptin-deficient mice leptin, they lose weight, but their blood pressure goes up. And so essentially what we have is a pathway whereby that probably explains a major component of the link between obesity and blood pressure. So as weight goes up, you get more fat, you get more leptin, you increase the signal through the melanocortin pathway, increasing sympathetic tone and increasing blood pressure. These patients have genetic disruption of this pathway, either leptin, its receptor, or MC4, and so they don't get the rise in blood pressure that you see with obesity. So we think this has kind of unraveled a major kind of system that's relevant for more common forms of disease. So after many years, a number of companies have now developed drugs targeting MC4R effectively. And the first place to give a drug like that would be in people who are lacking the ligand, or POMC. And this is data on that original patient with dramatic effects. And then we and others have been treating patients around the world with these drugs, patients with genetic disruption of this system. And they're doing very well. And these drugs are now licensed for some of these conditions and other conditions that we find if they map onto this pathway that provides us with the explanation to be able to treat these patients. Just a couple of kind of interesting, I think, scientific and clinical kind of stories just for the last bit of the talk, really. And this is around the fact that we're beginning to realize that we can find molecular explanations for clinical heterogeneity. So this comes about with a disorder that's been known about for about 50, 80 years in the textbooks, endocrine textbooks, and that's called, it's GNAS deficiency is what I tend to call it. Often people call it Albright's hereditary osteodystrophy or pseudo hyperparathyroidism, names which I could never remember as a student. But essentially, you get mutations in GNAS, which is the gene that encodes G-alpha S, which is part of GPCR signaling. Okay? And so what happens is you get a collection of clinical phenotypes, including obesity. We showed that the obesity is down to the link between G-alpha S and MC4R. But interestingly, many of the patients didn't have these textbook features. And the main point that I want to make is that when you're studying a disease that others have already studied and put in the textbooks, you sometimes have to do a bit more to overturn the dogma that exists. The other conditions I described were all described first by us, so we could sort of lay the ground for this is what we see. Here, what was interesting and challenging, and I think is an important challenge for anyone who follows up findings in human disease, is that the fact that patients didn't have all of these features led everyone to think that they don't have this disease. What they didn't think was maybe our understanding of the disease is too narrow, and actually maybe it needs to be redefined. And what we showed is that by characterizing all of these mutations in cells, by looking at their interaction with multiple different receptors, we could show they caused a loss of function, and we could link the number of receptors that are affected with the precise collection of phenotypes that the patient has. And I think this is what's going to happen in many different human diseases, is that so far we've tended to see only a subset of people with the disease who often have more severe forms of the disease. But as you do things like exome sequencing, and Baylor is world leading in this kind of field, you find more variation, and you find the spectrum of disease is wider than you thought. So I think that's going to be a very exciting area to come. And just now for the last bit of the talk, I'm going to focus a little bit on other behaviors. I'm very conscious a lot of people have interest in different aspects of neuroscience here. So these neurons in the hypothalamus project to different parts of the hypothalamus and other regions of the brain that affect behavior. And so what we're seeing is, as well as seeing obesity, we're seeing these other behaviors in these patients. So for example, SIM1 and OTP are two transcription factors that affect the development of the PVN, the paraventricular nucleus of the hypothalamus, which contains neurons such as oxytocin neurons. The people, and indeed the mice, have obesity, but also autistic type behavior, impaired social interactions. And so that's kind of quite clear. And interestingly, the prevalence of autism in our cohort is much higher than we'd expect. If you look at any autism cohorts, the prevalence of obesity is much higher than you'd expect. So there are clearly links between these two behaviors. Another group of conditions that we see go together with obesity is aggressive behavior. So here we see BDNF, brain-derived neurotrophic factor, well studied in the neuroscience field. It's receptor track B, and SH2B1, which is an adapter molecule involved in BDNF signaling, but also in leptin signaling. And patients with any of these genes disrupted have obesity, speech and language delay, impaired memory, and autistic traits, but also aggressive behavior. And the clue to that came with this, is that when I was calling up the patients and the families who had mutations, specifically in SH2B1, I was often getting a call back, not from their doctor, but actually from a probation officer or someone involved in their kind of criminal activity. So I thought, okay, it's a bit of a coincidence. Why are so many of these callbacks telling me about a disordered behavior in those patients, particularly in the males? So I thought, okay, well, it could be a coincidence. I mean, how can you sort of say, well, I've just happened to come across some slightly more criminal activity in this group than others. But I thought, okay, let me ask my colleague who made these mice, and this was Liangyu Rui in Michigan. And I asked him, look, this is a slightly strange question. Did you notice anything about the behavior of the mice? And he says, oh, Sadaf, let me tell you about the mice. What do they do? And he said, they're costing me a lot of money. And okay, why are they costing you money? Because I have to keep putting them in single cages, because if he puts them with loads of other mice in a cage, they kill the other mice. Okay, so I thought, okay, well, maybe there is a behavioral problem with the mice. So we then set out to test that. And then this is the data from the resident intruder paradigm, where you have a mouse in a cage and then you put another mouse in as an intruder and you see what happens. And basically, if you delete SH2B1 in the brain, the black bars, and then you test that the latency of attacks, the number of attacks, and the duration of attacks is high. These mice are very aggressive. When you restore expression in the brain, you normalize that aggressive behavior. So a clear link with this gene SH2B1 mediating aggression in these mice. There's an interesting sub-story is, I think we went to 13 different journals with this manuscript. And I think finally it got published in FASEB-J. And that, I think, was because people were just, editors, reluctant to believe or publish that something like aggressive behavior could have a biological basis. Okay. And I think it's incumbent on scientists to publish our research freely and discuss the challenges of our science and not let it be hidden by things that might seem difficult or challenging to believe. Other findings will kind of support this view in a second. So the other thing, and this is a collaborative work that we did with Yong and his team here, and it's great to see some of the team members who actually did this work are in the room, and this was on the serotonin 2C receptor. So this is a receptor that's on those POMC neurons, but many others have studied for a while, because drugs, for example, that treat depression and anxiety target the serotonin system, and actually the third generation antipsychotic drugs also target the system, and this may well be why patients treated for those drugs gain weight. And so what we found is loss of function mutations in people with severe obesity. And then here's Yong's work with the mouse model of one of those severe loss of function mutations, recapitulating the obesity, and then also showing that a behavioral phenotype, which is really supportive of our clinical observation of social anxiety. So these patients were anxious, but particularly anxious around people and meeting people. And we can see a similar level of anxiety in the mice, particularly with the social exploration test. So really fascinating to be able to explore these things in humans and in mice. And the last bit of work is really some unpublished work in collaboration with Yong's lab, where we were interested in a family of molecules that I think are really fascinating from a neuroscience point of view and a human behavior point of view. And these are the TRP channels. Okay, so these are cation channels that are widely expressed and really highly conserved across evolution. Okay, and they're there to detect sensory signals. So pressure, temperature, heat, smells, tastes. You can see lots of food here. So, we have trap channels for mint and menthol, but also for cool temperature. You have ones for chili and hot temperature. You have ones for garlic and oregano. So they detect our sensory signals and they convert them into electrical signals that can be picked up by the brain. We were interested in this one of these TRP channels, TRPC5, because colleagues in UT Southwestern actually had shown that the TRPC5 channel is located on those same POMC neurons, and it regulates the firing or the electrical activity of those neurons. So we became interested because we identified two families in which the boys had a deletion on the X chromosome that just takes out TRPC5, not any other gene. Okay. And they inherited those deletions from their mothers, who also had the deletion, who also had obesity, and they both had brothers who had the same deletion with obesity. So this looks like an X-linked disorder. What was really fascinating was the clinical phenotype of the children who lose TRPC5. So they had intense food seeking. They were hungry, but they would go looking for food, but they would also hoard food, which is not something I've seen before in humans. People don't collect the food and hoard it, like hide it outside the school to go and get later. They also had marked anxiety. They had autism and a clinical diagnosis of autism, and maladaptive behavior in response to sensory signals. So if they heard a baby crying in the street, they'd get very upset. That was too loud for them. If they smelled just some mild food being cooked in the kitchen, it would get too upsetting for them. Noise, volume would also be upsetting for them. And then they would get aggressive with their behavior. They also had persistent wakefulness. They were always awake as children for 2 hours with no problem. And interestingly, their mothers, who also had the deletion, had a history of postnatal depression. So we identified other missense mutations in this gene. And then with Yong's lab, we studied those in cells. And you can see that they caused a loss of function, impairing the electrical activity of that channel. And then we thought, okay, well, we're going to need to try to establish causality. We're going to need to establish this in mice. And so then Yong's lab made a mouse model of the severe loss of function human mutation. And basically, I won't go through all the data, but you can see it's shown here that the mice show an increase in food intake. Interestingly, they also do this food hoarding behavior, particularly if you change the environmental temperature. We think that this is a signal about food availability, and that some of this effect on food intake is mediated by those same POMC neurons. What is interesting is that the mice display many of those behaviors. The data on male mice shown here, increased anxiety. So there's the open field test. They're hanging around the outside. They don't want to go around and explore. Attack behavior, so aggressive behavior. They generally stay awake. And this is a fascinating test, the social interaction test. So reduced numbers of social interactions, preferring to interact with an object rather than a mouse. So really fascinating to recapitulate these complex behaviors in the mouse. And then I asked Yong a strange question. It's only because I know him well enough to be able to ask him this. I said, okay, this seems a bit mad, but I can't get over the fact that the two mothers have postnatal depression. And I've seen hundreds of patients with genetic obesity syndromes, and I've not seen that before. Could just be a coincidence, only two. But could you just have a look at the mothers? So Yong Chang is here. He's going to probably remember having to go and look and sending me a video of this. But essentially, we saw a striking phenotype in the mice that have this deletion and are then mothers. Okay, so best really illustrated by this photograph. So wild type mice, and then the mice with the deletion. And you can see that the nest is really poorly made and like scruffy, and the pups are scattered across the nest. And it takes a good while. If you place the pups at different distances, the mothers don't often go and pick them up and retrieve them the way they normally should. They take ages to retrieve them. The distance they spend away from the nest is quite far. So clearly impaired maternal behavior. And then the question is, what about depression? Well, in mice, you can test depressive-like behaviors with the forced swim test. You're in water and you're trying to see if the mouse will swim to the other end. And if they kind of give up and they despair, they don't tend to do that. So these mothers display depressive-like behavior. They don't want to swim to the end. And also they display reduced sucrose preference. So they don't like sugar, which everyone likes sugar, unless you have anhedonia. If you're depressed, you don't like rewarding things. And so the mice also display this depression. And so what's really fascinating is that this is present in the mothers, but not in male mice and not in virgin female mice. So that was a really beautiful set of experiments to really specifically show that it's in the postnatal period. And then this depressive behavior is linked with a failure in the rise of prolactin that normally occurs with suckling. So really fascinating to be first able to recapitulate all of these behaviors in the mouse. So then we homed in on oxytocin neurons in the paraventricular nucleus of the hypothalamus, which highly express this particular channel. And when you delete TRPC5 from those neurons, you recapitulate all of those features in the male and indeed in the female mice, including those effects on postnatal depression. And then using a virus, if you restore expression in those particular oxytocin neurons, you can correct all of those behaviors, incredibly also correcting the maternal care and the depressive behavior. So really very striking findings, linking this TRPC5 channel to a whole host of behaviors. I'll come to what I think it means, but I think this really opens up the way for understanding the function of oxytocin and potentially being able to treat a number of conditions. Importantly, I think this widens not only obesity, but also things like autism and also things like postnatal depression, which actually is the #1 cause of death in young women in the Western world. So women no longer die from hemorrhage or sepsis in the Western world around pregnancy, but they do die from suicide and postnatal depression. And there are actually no real mechanistic insights into what drives that. So I think it'd be fascinating to see if we could learn about the role of these oxytocin circuits in that setting. In the human brain, oxytocin neurons start off in the PVN, but they project very widely to other brain regions where you can find the oxytocin receptor being expressed. It's been challenging to target the oxytocin receptor and develop agonists or analogs because it's a very small peptide, but there are now chemists working with us on this. And interestingly, I think there's the potential, because oxytocin receptors heterodimerize with other GPCRs, including the serotonin receptors and serotonin 2C receptors, that you might be able to develop drugs that target both, given that there's a shared behavioral spectrum that's relevant. And people are beginning to develop drugs that basically target dimers effectively, which would become therefore a more potent kind of drug. And we're very keen to then work with Yong's lab to therefore give some of these drugs to the mouse models of the human diseases that we've developed as a proof of principle. And why do these things go together? Well, of course, as a human physiologist and a physician, it seems really intriguing. We've seen these things in combination, but actually the colleagues who work in flies and mice, they know this already. They've been studying these circuits for many years. So there are close connections between the neural circuits that regulate eating and aggression and reward and all these other behaviors. And they cause anxiety, reduce social interaction and aggression in flies and mice. And why do they go together? Well, I think they go together because they're innate behaviors. So these are behaviors that are fundamental for survival: eating, reproduction, aggression, defending your territory, your offspring, your mate, fear, anxiety, social interaction, and maternal care. So these are things that are hardwired in the brain, mediated by these neural circuits. And what we're finding is that if you have human mutations that disrupt the development or the function or even the maintenance of these neural circuits, you'll see the disordered behavior that comes from that. And I think that TRPC5 may well be a critical molecule that integrates a lot of these external and internal sensory signals to modulate innate behavior that's needed for survival. Okay, so really just to finish, overall, I think we've shown that obesity is complex and arises from the interaction of social, environmental, and importantly, genetic factors. And understanding that is quite important because it changes a little bit how we think about approaching specific patients, but also the societal challenge that we have with obesity. Genetic factors influence weight right across the spectrum. Thinness is also highly heritable. I haven't talked about our work in that area today, but we have now recruited a cohort of very thin people who are healthy but well can eat what they like and they never gain weight. I've never been that interested in those people, but I think they could tell us something that could help us treat people with obesity. And so we're trying to do that now. What we've shown clearly is that these genes influence appetite and behavior, and they can help us to identify mechanisms and targets for therapy. Relevance for clinical management. Clinical guidelines now recommend genetic testing of children with severe obesity worldwide. There are now mechanism-based therapies that can be used in these children. And really importantly, it helps us to remove the stigma from children and their families. And indeed, some of the families, people have tried to take the kids away and take them into care because they've blamed the parents for causing severe obesity. So one of the major benefits of this work has been to say, no, you can't do that. There's a biological cause for obesity here, and we need to support the families, not take the children away from them. But it's also important because I think of healthcare professionals also still retain considerable stigma towards people with obesity. So there's a job that we need to do to educate scientists, healthcare professionals, and others, and actually politicians too, about the biological factors that underpin our response to the environment. I finish by thanking my team in Cambridge, UK, my many collaborators, and of course, importantly, Yong and his lab with whom we've collaborated for 10 years now, and most importantly, the patients and their families. Thank you very much.
Fascinating. Thank you, Sadaf. So let's see if there's any questions here.
Oh, I've done something strange. Okay.
I can start asking
Yeah.
the first question. So I know that right now for the MC4 receptor agonist, so the strategy for the clinical trial is based on the biology that we know. So we are targeting those patients that we know that have genetic variants that may have impairment on important signaling. So what do you think if the drug could also be used more broadly for people that just develop obesity with hyperphagia, just like what people were using with GLP-1 agonist?
Yeah, so obviously there was interest in that. I think the early trials didn't show much of a response in common obesity with the MC4 agonists. I think it's yet to be seen whether there may be subtypes of people who might respond. And for example, one way in which you might do that might be not just as necessary to drive weight loss, but to drive weight maintenance. Because when people lose some weight, what happens is their leptin levels fall and you kind of increase signaling through this pathway. And giving back leptin or something downstream of leptin at that point might help prevent weight regain. So I think that's probably where more likely the use could be. A number of companies are now developing different types of drugs, including orally available drugs for the system. Yeah.
The data that you showed with people actually eating more of the fat, and as you said, like that's happening in minutes. Do you have any thoughts on how leptin, how exactly that pathway is regulating at some taste receptors or some, how exactly they're choosing that?
Yeah. So we don't, I mean, I think it must be something in the mouth because it's very, very quick. And so I think that's something that needs to be figured out, actually. In fact, people in this room who need to take that up and figure that out because it's the total amount of meal consumption is the same. So, and it's too quick to be, the meal is over in 30 minutes. So it's too quick for the gut hormones to go and then tell the brain to eat this food. So it's something very immediate that's actually picking it up and telling you this is incredibly rewarding, eat more of this. And of course, we've taken great care to make sure they can't tell which is which. We make everybody test each one before they have it, so it's not they're going to the nearest one. We swap them around. So we've tried to address any kind of confounding variable. So I think it's very clear that it's the fat that's being detected. It's the fat content that's being detected. We know we've done other studies with my colleagues in Cambridge to look at how quickly the brain detects fat. So if you're lying in a scanner, MRI scanner, you can feed people through a tubing milkshakes of different concentrations of fat. And you can also check them for mouthfeel, how slippery they are. When you control for that, the high fat milkshake causes very acute activation in the brain when you're just squirting in 1 ml into the mouth. So there is something about fat that we can detect very quickly.
Thank you for a very enlightening talk, inspiring to have a metabolic reactive surgery. And, we're still very early on. We have very just a handful of operations that we can offer a large population of patients. And our results are not always successful. There are patients who do well and others who don't. And we still don't have enough information to clearly select the right operation for the right patient. And in addition to Dr. Sisley's, I'd like to build on that. We have noticed that a lot of our patients' behavior and food preferences change. But my problem is with those patients who don't respond well to surgery. I wonder if you and your group have worked with surgeons to maybe try and help them select better, which operation for which patient.
Yeah, great.
Can I ask you to repeat the question?
Yes.
Because there is a people.
Sure, great. So the question was about the response to bariatric surgery, the variation in response, and also the subgroup of people who don't respond, and whether we can identify them better. So really important clinically driven questions. We've done one piece of work, and then others are doing some work on exactly those questions. So what we do know, for example, is that the response to bariatric surgery, if we take one procedure, Roux-en-Y bypass surgery, is variable. And also that genetic factors influence that variability. So a very large study that we were part of, led by Lee Kaplan from MGH, whereby they took 1,000 people who'd had bariatric surgery, and they looked at the variation response, and they also had DNA on those people, and they could show that it was heritable. Okay? So people who happened to be related or distantly related had similar patterns of response versus, say, for example, a husband and wife who were not biologically related. So the response to the surgery, and indeed to any weight loss intervention, is heritable. We haven't got specific genes that mediate that. What we know, for example, is that MC4R patients respond as well to Roux-en-Y bypass surgery if they are heterozygous, 1 faulty gene, but not if they're homozygous. They don't respond well. And indeed, the homozygous mice, Lee actually did the bariatric surgery on those mice, and they don't respond to bariatric surgery. So you need an intact system for response. So I think what's likely is that people who have problems with this system in the brain may be the ones who are not responding well to the surgery. But at the moment, we don't know enough about which particular people they are. People are doing that. So a number of colleagues around the world have collected DNA on the same people on whom they've done the surgery. And hopefully, I think by pooling those cohorts, we may have enough to get those kind of insights. But I agree that it's a major question. And at the moment, people are also trying various treatments after failure of surgery, and then trying to see if we can better stratify those patients.
Wow, that was a really fascinating, inspiring talk. I'm grateful that you shared your time and knowledge with us. I'm curious if any of your work has focused on kind of the other side of the energy homeostasis, looking at energy expenditure and any maybe genetic differences that underlie maybe adaptations to increase food consumption that maybe make people more likely to eat despite environmental pressures leading other people to become obese.
Yeah, absolutely.
And how that circuitry may correspond to the
Right. So a couple of questions in there. So big questions. So genetic factors, question was about genetic factors influencing energy expenditure in the population, and then more broadly about potentially whether those and or other factors might explain leanness or people who remain thin. So two things. So if we talk about, there's clear evidence that aspects of energy expenditure are heritable. So basal metabolic rate is highly heritable. So there are genes that will influence basal metabolic rate. We know some of them, not so many. So one of the genes we've studied is something called KSR2, where the people and the mice develop severe obesity, and the predominant defect in both is actually energy expenditure. Basal metabolic rate is lower. And indeed, the mice don't survive well when exposed to the cold. Okay. But most of the genes that we found affect predominantly appetite. Basal metabolic rate is heritable. Some very nice studies from the Quebec family study show that the physiological response to exercise, a fixed amount of exercise, is also heritable. So how many calories you burn when you're given a fixed amount of exercise is also heritable. We don't actually know many of the genes for that. So I think there is still yet work to be done to understand that aspect of the system and its variation. Some of that, I think, will come by studying thin people. Okay, so that was partly our motive to recruit a cohort of thin people. We were able to, they don't really come forward for medical attention, but people who are thin, I'm talking about a sort of BMI 18, 19, so really pushing it to the thin end of the spectrum, but are well, don't have eating disorders, don't have chronic medical conditions, psychiatric conditions. And fascinatingly, we have genotype data on 1,000 of them. We've got a total cohort of 3,000. And what we see is about 20–25% of them report not being that interested in food. So that's probably the major driver for them. About 75% of them report being pretty interested in food, can eat what they like, and they still don't put on weight. Okay, I'm determined to find out what's wrong with them and how that works. Okay, and I think that will be really fascinating and teach us some things. Part of me was thinking, okay, but yeah, you don't probably eat that much, do you? And so I started bringing some people in for physiological studies. Just an example of our prototype first person who came in, the young guy, 27, weighing 42 kilos. Okay, and really asking me to help find some way to help him gain weight. I thought, oh, I really feel for you, my friend, but let's try and see what we can do. So we kept him in for a study for 10 days, okay, in our unit under close supervision, and measured everything before and after, overfeeding by 100%. Okay, all of the extra calories came from fat. Okay, and nothing changed. He didn't gain even half a kilo. Okay, his metabolic rate didn't change, his bloods didn't change. The only clue was excess sweating. Okay, so is he somebody who can lose extra calories through thermogenesis? Okay, anecdotally, we do often know people who are always walking around in a t-shirt in the cold weather, or who are always feeling very hot and can often be slim. I have people in my own family who are like that, and I think that there may well be genetic variants explaining why some people can probably burn more calories and stay slim. We haven't really found them yet, but I think we're getting close.
Yes, very inspiring. I think this type of research really provide guidance for us. Very say your game function. My question must have other Steve, they found that starching what's your
I think you might be missing MC3 and MC4R. So gain of function MC4R do protect against obesity. MC3R do not affect obesity. MC3R affects body composition and the timing of puberty. So I think that may be what you're referring to. So it looks like different functions for the two major brain-expressed melanocortin receptors. Some people did find MC3R mutations in children with obesity and then were thinking they were causing obesity. We weren't really that convinced from our own data, but I think it now does look clear that it's MC3R probably drives excess calories towards body composition and the timing of puberty, whereas MC4R is clearly driving food intake and weight. No, they're the same ones. Yeah.
Okay, I'm gonna give the last question to our audience online.
Okay.
So they're all over the world. Some of them actually midnight right now. So there are many questions, but I can only pick one, which is my favorite one. What about sex differences in the frequency of the mutation of all these genes that you mentioned?
Right. So sex differences in the frequency of the genes. So most of the genes that are on the autosomes, the frequency is the same. So in males and females, so MC4R, leptin receptor, the frequency is the same. There are a subset of genes that are on the X chromosomes. Okay, TRPC5 at the very end that we mentioned, serotonin receptor is also on the X chromosomes. And there, most of the people that we see affected are actually female. And I think because being male, you would effectively be hemizygous or have no functioning copies, it's probably too severe a phenotype. So far, that's what we have learned. We don't really see any evidence of a preferential male-female distribution for genes other than the ones that are X-linked.
All right. Thank you. Once again, let's all thank Wonderful talk. And thank you everyone for coming. So, yeah, we'll meet you to your next.